guys, welcome back to the channel. So in this video we'll discuss more about microbial growth and kinetics under biochemical and bioprocess engineering. So previously we discussed about its reactions and some of its terms involved in this process. So we'll just continue from there on. So let's get started with this. So starting with this, we talked about this in the later part of the video, in the previous one. So we talked about this, which was the normal graph phase for the microbial growth. So we talked about all four terms such as lag, exponential, deacceleration, stationary, and death phase. So we made it out clear that how it obtains and what are its significance and how this sort of curve has obtained. So moving on from there on, so we'll talk about each one of them in detail. So starting with the first phase, which is the lag phase. So every microbial reaction or microbial growth phase starts with the lag phase. So let's just talk about some of its facts and how it is interdependent or dependent on the lag phase. So the Enterobacter aerogens. So this is a bacteria which, when grown in glucose and phosphate buffer medium, increases lag phase as the concentration of Mg2 plus is increased, which is an activator of the enzyme phosphatase. So the concentration of Mg2 plus or the magnesium ion concentration is very important in extending or decreasing the length of the lag phase. So more the concentration of Mg2 plus, the higher or the lengthier the lag phase is, because Mg2 plus is an activator of the enzyme phosphatase. Phosphatase is involved in the increasing of length or making the process lengthier. Alright, so Mg2 plus concentration is very important, or the presence of Mg2 plus concentration is important for the microbes or bacterium to adapt to situations and thus start the growth phase. Also secondly, so the age of the inoculum culture has a strong effect on the length of the lag phase. So definitely a new culture will have a different growth curve, whereas the old culture kept in a couple of hours or a couple of days, so will have a different sort of curve. So age of inoculum matters a lot, and also inoculum stored like more than a week or like more than 4 or 5 days won't be as effective, because most of its bacterium would be dead, or we will observe some sort of contamination in that. So basically it's preferred that a 24-hour grow culture should be used, or somewhat that, but not a very old culture should be used. So also that affects the lag phase and also the entire growth curve. So talking about the third point, which is the Diauxic growth, which is the most important point to talk about. So basically the Diauxic growth occurs in two carbon source. Alright, so what is it? So let's just get through this. So after one carbon source is exhausted, there is a shift in the metabolic pathway in the middle of the growth cycle. So this is Diauxic growth occurs in place of microbes. So let's say the one carbon source is glucose. So when the glucose source or the one carbon source is basically the nutrient source. So when this nutrient source, which can be glucose, when it gets exhausted, so it moves to a different carbon source, which can be lactose or any other carbon source in the middle of the growth cycle. So when the bacteria moves into the lag phase, or from lag phase it moves to the exponential phase, if there is an exhaustion or scarcity of some sort of carbon source, let's say glucose, so then at that point it shifts to a different carbon source at that point of time or in the middle of the cycle to continue its growth. So that we know is the Diauxic growth. So it carries its growth in two different carbon sources in the same cycle. So it does not let the cycle to stop. It does not stops growing. It does not lead to death, but it remains active by shifting in its different carbon source. So this was about lag phase facts. About more. So let's just move to the second growth phase, which is the exponential phase. So this we'll talk about exponential phase, which is one of the most important phase considering the microbial growth. So the exponential growth phase, so since the nutrient concentrations are large in this phase, so the growth rate is independent of nutrient concentration. So as the bacterium has just adapted to the conditions and the nutrients are present in heavy quantity because the bacteria microbes are very new to the environment, so just they are adapting to the environment in the lag phase, and as they have adapted to the environment they start growing, and thus they move to exponential phase. So adapting to conditions and moving to the next phase, which is the exponential phase, does not decrease the nutrient concentration because the nutrient concentration is at large because they have just started multiplying. So the starting of multiplication does not have anything with nutrient concentration being less. So they have nutrient concentration in abundance. So this time the growth rate of the microbes or the bacterium is independent of nutrient concentration because that is present in abundance. So there is lots of nutrient right now in the exponential phase. The nutrient concentration decreases when we move into the stationary or the deacceleration phase. Also, the exponential growth rate in first order is it is given by dx by dt equals to u net mu net x. So mu net, as we know, is the specific growth rate, net specific growth rate. In this, x equals to x0 and t at time 0. Also, after integrating the this particular term, we get ln x by x0 equals to mu net t, or we can represent it just by writing this, which is x equals to x0 e to the power of u net t, in which x and x0 are the cell concentration at time t and t0. So this is how we represent exponential growth phase at first order. All right, so this is the particular result we get. So moving on with, so let's talk about more and how it affects how some more of its features. So let's just talk about the doubling time, cause here the microbes multiply at a much faster rate, at an extreme rate. So this is the fastest growing phase the microbes are into. So this that's why we are talking about the doubling time. So the time required to double the microbial mass. So the bacteria are going extremely at extreme rates. So this is the point where we'll calculate the time required to double the microbial mass. So we just calculate the time, how much it takes to double its mass. So the exponential growth is characterized by a straight line on a semi-logarithmic plot of ln versus t. So as we know, the exponential growth is just this, right? This part, the down part, is the lag phase, and this straight line is the exponential part, and thereafter we have the deacceleration and the stationary phase. So this part is the exponential growth, which is pretty straight versus time, as we know. So the doubling time is given as, so this is tau. So this particular term is represented as tau. So tau d equals to, so this is d represents doubling time, and tau d equals to ln2 by mu net. So ln2, we know the value of ln2 is 0.693 by net specific growth rate. All right, so this is the way we can calculate the doubling time in an exponential phase. So this is only prevalent for exponential phase because the growth rate is pretty much high. Also, calculating the doubling phase based on number of cells and net specific rate of replication. So basically there's not much difference. So u net gets replaced by mu r. So mu r is net specific replication rate. So net specific rate of replication. So while calculating the doubling time based on the number of cells and the net specific rate of replication, so we'll just replace the net specific growth rate by net specific rate of replication when we are considering the net specific rate of replication on cell number. So this is tau d dash is the doubling time based on replication rate, and this is tau d is the doubling time based on a net replication net growth rate. Yeah, this is the net growth rate and this is the net replication rate. All right, when we are talking about this on the numbers. So this is a pretty easy one, I guess. So moving on with this, so we'll talk about this thing, which is one of the most important topic, which is the monod kinetics. So how monod kinetics comes into play. So as you see two figures here, so let's just talk about the first figure first. So this is where the specific growth rate plotted respected to initial substrate concentration in a batch system. So as we know the specific growth rate, this is plotted according to the initial substrate. So the substrate starts from here or maybe here. The substrate concentration is pretty high. As you can see, it's high, it's high, it's high, and a point comes where it reaches the saturation. Let's say this is the saturation point. Or let's just say come into the second graph where we see Umax, which is the saturation point, where the substrate concentration cannot increase further than this, and this is the saturation point. Whereas this is the half of saturation point, which is denoted by Umax by 2, and whereas this saturation point is denoted by Umax. Alright, and from there we can calculate mu. So mu equals to mu max in brackets S by S plus Ks. So S is the substrate concentration and Ks is the saturation. Alright, so moving on with this, I'll be talking about the monod kinetics entirely in much detail. So let's just talk about the stationary phase, like how it is important. So before like moving on to stationary phase, there we studied about one more phase, which is the deacceleration phase, which comes before stationary phase. So deacceleration phase is nothing much but something that is just before stationary phase and just after exponential phase. So this is the deacceleration phase is the smallest phase, whereas the bacterium, the growth rate decreases a bit and our death rate rises a bit. So to stabilize that point, deacceleration phase is, we name it as deacceleration phase, where the growth rate decreases a bit and the death rate increases a bit. So there we observe a slight instability, and thus moving that very small period, we call it as deacceleration phase. So after the deacceleration phase, the microbes move into the stationary phase, where the growth and the death rate is almost the same. So talking about this, so during the course of the stationary phase, one or more of the following phenomena may take place. So total cell mass concentration may stay constant, but the number of viable cells may decrease. So the thing is that the X remains constant and N may decrease. So as we have studied in the previous video, if you don't remember, you may watch the previous video. Just give it a watch. So the total cell mass concentration, which is denoted by X, all right. So total cell mass remains constant or may stay constant. So the concentration per area remains constant, whereas the number of live cells may decrease or number of viable cells may decrease. So the number of cells is denoted by N. All right. So the number of cells decreases, whereas the cell concentration remains the same. Whereas this happens, whereas this condition is opposite when we talk about lag phase. So under lag phase, the number of viable cells may remain constant, whereas the number of cell mass concentration increases. All right. So the number of viable cells may remain constant or slightly increase, but the cell concentration increases a lot while in death phase, while in lag phase. Whereas in stationary phase, the cell mass concentration remain more or less the same, whereas the viable cells decreases, and viable cells decreases means it leads to increase in death rate or increase in KD. All right. Also, cell lysis may occur and viable cell mass may drop. All right. A second growth phase may occur and cells may grow on lysis products of the lysed cells. So cryptic growth, which we call it. So after cell lysis may occur and viable cells may drop, so it leads to the increase in dead cells. So at that point, a second growth phase may occur and cells may grow on lysis products of the lysed cells. So this particular point means that after the death of the cells, after some of the cells which die, so the live cells which are present or which are already present in that, which are alive, feed on the dead cells. So the live cells, for their survival, now feed on the dead cells which just died or which died during this process. So the live cells or the viable cells now feed on the dead cells to survive. So which we known as the cryptic growth. So cryptic growth is just simple, so that the which we also know as the second growth phase or the cryptic growth. So under cryptic growth in stationary phase, so the viable cells, viable or the live cells which are present in this medium or the environment survive by feeding on the dead cells. So coming to the last point, which is the cells may not be growing but may have active metabolism to produce secondary metabolites. So these may not be actively growing, but they have an active metabolism to produce secondary metabolites such as different sort of sugars and all. So secondary metabolites are produced as a result of metabolite deregulation. So this is the point. So this is the phase where we see formation of secondary metabolites. All right. So we see the formation of secondary metabolites in stationary phase. So moving here, so let's just talk about some of its conversions and some of its denotations. So this is the phase where we see endogenous growth, endogenous metabolism, pardon, caused due to some deaths. As stationary phase, the cell catabolizes cellular reserves for new building blocks for energy producing. So this is the phase where we see endogenous metabolism, we see cryptic growth, we see a lot of production of secondary metabolites, because this is the phase where the cells are pretty weak and they need something or the other to survive. So these lead to the formation of secondary metabolites as we studied. Also the conversion of cell mass into energy or cell death. So conversion of cell mass into energy or cell death is given by x equals to xso e to the power of kd by t. So this was the term. After integration we found this. So in this kd is the first order constant for endogenous metabolism, and xso, so this is the xso, is the cell mass concentration at the beginning of the stationary phase. So xxo is the cell mass concentration at the beginning of the stationary phase, and as we know, at stationary phase, S0 and UG0. So these are two time periods, x and xs0, at two time periods, as we know, S0 and S1, ST, or we can say. So moving from there on, let's talk about the last phase, which is the death phase. So talking about the death phase, so the rate of death usually follows first order kinetics. So this is something that we need to remember, that almost all of the phase generally follows first order kinetics. All of the three that we studied, that like exponential, stationary, and death. Lag phase is not something that we like count in this. So cause lag phase has nothing to do, cause it is just a phase where the bacteria microbes take time to adapt to the situation before they start growing. So in this under death phase, it is given by n equals to ns e to the power of minus kd dash t. So in this case we don't use x. So x in every case is the cell concentration, cell mass concentration, and n is the cell number. All right. So in this n or the ns. So ns is the concentration of the cells at the end of the stationary phase, and kd is the first order death kinetics. So this is the concentration of cells at the end of the thing, or we can say this is the number of cells at the end of the stationary phase, and we can just plot of ln n versus t, which yields a slope of minus kd. So we will obtain a slope like this for death phase, which has we'll obtain a graph of death phase like this, which has a slope of minus kd. All right. So and it is which is given by this. So this was about the death phase. All right. So talking about the last term which we have in this video for is the yield coefficient. So talking about the yield, how much the bacteria multiplied, how much was the product released, how much the dead cells caused, or releasing of secondary metabolites and everything. So yield coefficients are defined based on the amount of consumption of another material. So basically it basically means that the amount of nutrients that microbes consumed would be equal to the amount of product that should be extracted, or that should be like more or less equal. All right. So it says that it is based on the amount of consumption of another material. So the amount of consumption should be equal to the amount of extraction. But let's see if it's equal or not. We need to, like, there are many sorts of numericals through that we can find, and it is yield is denoted by x by s. S is the substrate and x is the cell mass concentration, which is equals to minus delta x by delta s. All right. Also substrate consumption is denoted by this, which is assimilation into biomass, assimilated into an extracellular product, growth energy, and maintenance energy. So this is how we denote the yield coefficient. So let's just keep this video till here. Hope you like the video. Stay tuned for more, and thank you for watching this.